Next we have Aubrey Annis speaking about multiple testing correction in biobank data.
Hi, my name is Aubrey. I'm in the Abacasis lab in the biostatistics department. And you've heard a couple times mentioned already today permutation methods. And so that's what I'm going to talk about, our work with permutation methods and how MGI was instrumental in forwarding that work. So most of us are familiar with single genome-wide association studying multiple testing correction. We have a current genome-wide significance threshold that usually rests at 5 times 10 to the negative 8th. And the origins of this threshold lie in multiple testing correction for approximately 1 million common variants across the genome and a single phenotype. So we've come a long way from this original setup, and to illustrate, we can think about MGI, and this is talking about Freeze 2 of MGI, so we have even more variants now with the new Freeze coming out, but in Freeze 2 we had approximately 32 million variants that were testing across the genome and about 1,700 phenotypes. And so we know that the more tests that we perform on data, the more likely it is that we're going to have false positives occurring by chance, and so we need to have some sort of correction for this large biobank situation. Other complicating factors include that we also are testing across common and rare variants with different allele frequencies, and that we have correlation among our electronic health record phenotypes. So Lars showed some of the phenotypes that we're testing, and many of them have relationships to each other and are highly correlated with each other. So these are other complications that we have to think about when we're looking at doing multiple testing correction in this biobank data. So I'd like to present a single phenome-wide permutation method that we've been working on for a couple of years. And so on the slide, you can see a diagram of primary data. This is just the data that we would get from our MGI cohort with genotypes and genotypic covariates on the left, and phenotypes and phenotypic covariates on the right. And the idea of the single permutation method is that by doing a permutation within an entire biobank, we will have enough randomized data to calculate false discovery rates. And so the permutation proceeds by shuffling phenotype vectors among participants. This breaks the genotype-phenotype associations and provides us with null data from which to calculate false discovery rates. From this stage, we go on to perform genome-wide association studies across all of the phenotypes in the biobank. We perform some clumping to find independent signals. And finally, we can estimate FDRs based on the number of permuted independent hits divided by the number of primary independent hits. So I'd like to show you the utility of this then in MGI. So this is a heat map that's showing our different false discovery rate estimates in our MGI data across 1,700 phenotypes. It's partitioned by minor allele frequency and by p-value category. And so we can see that a couple things here. One is that, of course, our false discovery rates are going to be varying by p-value, and the more significant p-value that we have, the fewer false discoveries, as we'd expect. But we also see that it's varying by allele frequency. And so as we go down the plot into the rarer variants, we can see that we have a higher false discovery rate probability. Other things that can affect our false discovery rates in these different categories are the phenotype correlation specific to the data set being analyzed, the ancestry of the participants, and also the relatedness amongst the participants. And so the kind of main takeaway here is that these false discovery rates are going to vary by a lot of different features, and those features are specific to the data sets, and so we really need to get false discovery rates for the specific biobank that is under consideration. So I'd briefly like to consider the difference between false discovery rates and Bonferroni and multiple testing correction. So what I've highlighted right here is a traditional Bonferroni threshold of 5 times 10 to the negative 8. And you can see as we look down our minor allele frequencies that we really have a high proportion of false discovery rates among rare variants. And if we look at the type 1 error and type 2 error, this is estimated from simulations that we did. The type 1 error is extremely high, and the type 2 error is extremely low. On the other hand, if we were to do a traditional Bonferroni correction across 1,700 phenotypes, which would just be dividing by the number 1,700, then we would come out with a p-value threshold of 3 times 10 to the negative 8th. So in this situation, we have the opposite problem where we have very low type 1 error and we're getting good signals, but we have high type 2 error. And so what the false discovery rate allows us to do is, instead of just taking a stripe across these different p-value categories, we can take this triangle area here. And it allows us to balance between our type 1 error and our type 2 error at approximately 4% and 20%, which is what we generally aim for as statisticians. So finally, I'd like to think about how this can be useful in interpreting FDRs in a scientific setting. And so I've presented here 2 different genome-wide association studies from our MGI data, and they really look extremely identical if you look at the peaks. We have corneal opacity and disorders of the cornea and acquired hemolytic anemias. And probably from a single GWAS, if we're looking at these, we think these are equally reliable signals. However, if we calculate the false discovery rate based on the minor allele frequency and the p-value, we can see that they have very different false discovery rates, with the top signal having a 4% estimated false discovery rate and the bottom have a 72% estimated false discovery rate. And what we actually found when we dug a little deeper into these signals is that, first of all, the top signal is replicated in UK Biobank very strongly, while the bottom signal is not. And these also fall well within clinical parameters that we'd expect for these phenotypes, with corneal opacity is known to have a strong genetic feature, whereas acquired hemolytic anemias are usually environmental features. And so with biobank data, we recommend that all biobanks report false discovery rates along with p-values and minor allele frequencies. So we've shown here how this is very effective in MGI. It's also effective for you as researchers to use this MGI data yourself, and it also sets a standard for other biobanks around the world. This paper is coming out soon, hopefully in Nature Genetics. We just returned our reviews to the reviewers, so you can look forward to that, but there's a preprint available on Research Square now. Thank you.